The overall goal of this experiment is to observe the real-time protein trafficking in primary neuronal cells, such as GLUT4 in hypothalamic neurons, with minimal damage.
This method can explain key questions related to neuronal regulation of insulin signaling and metabolism, such as in the case of type 2 diabetes mellitus.
The main advantage of this technique is that the researchers can isolate primary cell cultures and to observe the drug-induced effect in real time.
The implication of this technique is to the therapy of type 2 diabetes or brain-related insulin resistance, because now we know how does insulin signal being altered by the chemokine CCL5 in hypothalamic neurons.
To begin this procedure, coat the culture dishes with poly-D lysine 1 day before culture. For a 6-well dish, add 1.5 milliliters of poly-D lysine at 0.05 milligrams per milliliter to each well. The next day, remove poly-D lysine and wash the dishes twice with 2 milliliters of DDH2O. Then, fill the 10-centimeter Petri dishes with 20 milliliters of wash medium and keep them on ice. Fill the 15 milliliter tubes with wash medium and keep them on ice. For brain tissue isolation, sterilize the platform, dissecting microscope, and surgery tools with 75% ethanol to avoid contamination. After that, cut around the umbilical cord area to isolate the pups without damaging them. Next, remove the head of each pup and secure it in place using the fine